How about that? All right,
Let's
got it. Okay, so what I want to talk about is um, the process of doing a cure and what components you need to get your students to a hypothesis, et cetera, and like how does the cure work? Um, and so starting building with what Chris and Jess both said, the key thing is that we all need to start with some three big areas, which is we need to get information, foundational biochemical knowledge, right? What are the types of non-covalent interactions? What are their strengths, et cetera? We need some basic concepts of enzymology. What's an active site? How do proteins fold? Do they have to be folded to be active? You know, some of these very basic fundamental biochemistry things, but they're really important for students to be able to develop a hypothesis to have some familiarity. Um, we also have all the MDH background, like what Chris and Jess said, but even more. There's a lot more detail in the published literature and particularly in the essays in biochemistry articles that kind of pull together a lot of the literature. And then there's properties of MDH itself. So what about the sequences? What about the structures? And students need some familiarity with that in order to develop their big picture idea. Okay, so the start is what is the big picture idea that they want to ask a question about? And this big picture idea, you could let them choose from anything, but I think our next two speakers are going to encourage you to kind of you yourself first select an area from everything possible the students could study about MDH and narrow it down to some subset. But then within that, students develop a big picture idea, and then they discuss this big picture idea with the instructor, with their peers. In my class, and I think in a lot of others, I have them do a one slide elevator pitch of their big picture idea, just to get them to kind of pull it together and then get some feedback. And then once they get a little bit of feedback, they circle back and look back to their foundational knowledge, the MDH information, properties of MDH, to revise their idea and eventually settle in on a formal hypothesis, right? And the hypothesis should be um, uh, a prediction, right? A hypothesis, a particular question that they want to answer. Um, And using that hypothesis then could be something like, I think that one of the interactions Jess just talked about, I think this is crucial for the binding of the cofactor. Okay, so that could be a hypothesis. Then you could make a prediction, well, if I change that residue, now the cofactor won't bind as tightly. So students need to get from a big picture idea, we're studying the cofactor binding site, to a hypothesis, this residue is important, to a prediction, changing this residue will reduce binding, for example. And then that leads to them to propose an experiment. How can we test this hypothesis with an experiment? What experiment are we going to do? And this is where obviously you have to think about it because it has to be an experiment that you're prepared to do with the students, that you have the right equipment, that you have the supplies and everything. So some direction in this area can be really helpful. Okay, so that's the big part of developing the hypothesis. And one thing I think people will talk about a little bit later is this takes a lot longer than you think. The students have to synthesize a lot of information that they're not familiar with and expecting them to do it in a couple of hours. You and I could probably do it in a couple of hours, but they need like a few weeks to do this well and synthesize the information. And then once they've done it, this is a more traditional biochemistry lab, right? We're going to perform the experiments to test our hypothesis, analyze the results, and draw conclusions. And then usually as part of the MDH CURE, students present their results. Could be a report, but could also frequently, many of us have them do a presentation, a poster or talk or some kind of presentation of their results. And then they need to think about how would you revise the hypothesis based on the results that you obtained after you did your experiments. And so those are the key elements of the cure that students need to be aware of as they work on this MCC project. And so I'll stop there and take any questions, and then we'll get some applications of what kind of projects people could work on.